Hello guys, welcome to the knowledge domain, the concept of life sciences and biotechnology. In this lecture, we are going to learn the cell junctions. There are 200 types of cells in the vertebrates based on the visible difference. The cells can be tall, columnar, squat or pivotal, flat or the squamous. So till now, we know that the cells are the structural and the functional unit of the living organism. It is so simple to understand. But how these cells, they organize themselves to make the species like mammals, birds, reptiles, whales, and whatever animal we see around us. How they communicate with each other, how they form communities and organize themselves into cooperative assemblies to perform specific functions called the tissues. We have discussed in the previous lecture of the ultrastructure of the cell, where there are 4 types of the tissues in animals. It is the epithelial cells, these are the epithelial tissues, muscular tissues, neuronal tissues, and the connective tissues. So a tissue is composed of cells, certainly, and the extracellular the material that is called the extracellular matrix. So there is a cell-cell communication and the cell extracellular matrix communications, or cell-cell junctions, or the cell extracellular matrix junctions. And this tissue, it provides the flexibility and the mobile nature of the animals. The force generated in the cytoskeleton of a cell, it transfers it to another cell or into the matrix. These are the characters of the extracellular matrix or the components present in the extracellular matrix. They provide the top and the flexible characters found in the tendons in the dermis, hard and the dense nature of the bone, resilient and shock absorbing character found in the cartilage, soft and transparent characters found in the vitreous of the eye. The collagen that forms 25% of the total protein mass in a mammal is a major constituent of the extracellular matrix. So this collagen is connected to the cell through a protein called the fibronectin. Another protein, another polysaccharide, glycosaminoglycan, that is negatively charged. It gives the resisting compression of the tissues. So as the epithelial cells have almost all types of the cell junctions, we will take epithelial cell as an example in discussing the various junctions. So epithelium lines the external as the internal lining of all the organs and the tissues. This is an example of the gut wall, where we can see there is a epithelium towards the lumen of the gut, and there is epithelium on the outer side of the gut wall. Both epithelium sits on a connective tissues, and in between them there is a circular muscle fiber layer and there is a longitudinal muscle fiber layer. These epithelium, they secrete specialized products like hormones, milk and the tears from the glands, absorbs nutrients in the intestinal epithelium, detects signals like the light in the photoreceptor, the rods in the cones found in the retina, detects signals like sounds in the auditory hairs of the internal ear epithelium. Cells join together into an epithelial sheet that creates a barrier. In case of the multicellular organism, a kin to the plasma membrane of the single cell. Both creates the barrier. So the epithelial sheet takes up nutrients, export waste. It has the receptors for the environmental signals. It protects the organism from the invading microorganisms and prevent the fluid loss. Epithelial sheet is polarized and rests on a basal lamina. The apical surface is free and exposed to the air or to the watery fluid, whereas the other sides of the epithelium that sits on the connective tissue called the basal lamina is the basal surface. The collagen found in the basal lamina is type 4 that provides the toughness to this epithelial sheet, and the protein that provides the adhesive characters is the laminin. So this is a tight junctions. Tight junctions make the epithelium leak proof, and it is this tight junction that provides the polarity to the epithelium. It separates the apical and the basal surface. As there is a tracer molecules, they were put on the lumen, they do not cross the tight junctions. From here it is confirmed that there are two surfaces or the two poles and both are separated by the tight junctions. This is an electron microscopic image where you can see the tight junctions. The proteins found in the tight junction are occludins and the chlorines. They are the character. So they provide, they make, or they have created the different environment across the junctions. Because of this tight junction, the pumping functions is possible to the epithelium, like there is a proton pump present in the stomach that secretes the H plus into the stomach lumen. There is a one flow of the nutrients and the waste. It provides the mechanical attachment as well as the chemical communications. So this is an example of the one-way flow of the nutrients. This is the intestinal lumen in case of the small intestine, where the carbohydrate is already digested, and the result in the monomer is the glucose and the galactose as well as the fructose. We take salts in our food, so there is the presence of the sodium. So glucose, galactose, along with the sodium, by the sodium glucose transport enters into the epithelium or the epithelial cell, cytoplasm. So the sodium glucose transport, SGLT. This sodium is entered into the extracellular fluid found on the basal surface of the epithelium by the sodium potassium ATPase, where the glucose and the galactose, they move by the facilitated diffusion. Into the basal surface, into the extracellular fluid, and finally into the plasma, where the fructose by the GLUT5 it enters into the epithelial cell cytoplasm, and like the glucose and galactose, the fructose also by the GLUT2 it enters into the extracellular fluid, finally to the plasma. So because of this tight junction present here, the sodium glucose transport as well as the GLUT5 cannot move to the basal surface, and the sodium potassium ATP as well as the GLUT2 it cannot move to the apical surface, because as we have discussed in the plasma membrane, the phospholipids and all the lipids as well as the proteins found on the plasma membrane, they have the lateral movement. So if this is present on this apical surface, they have the ability to go to this surface based on the fluid mosaic model of the plasma membrane. But tight junctions is preventing the lateral movement of the lipids and the proteins found on the plasma membrane across it. And because of these characters of this tight junction of preventing the lateral movement of the lipids and the proteins on the plasma membrane, it gives the polarity to the epithelial cells. So before going into the next part of the cell junction, that is anchoring in nature, the cytoskeleton that we have an introduction in the eukaryotic cell description, we will discuss it. So we know that there is a microfilament, the smallest one, or the actin. Then there is the intermediate filament and the microtubules. So the three types of the cytoskeletal elements. So the actin, the monomer is the G-actin, that is globular in nature. The small G-actins, they are soluble. And when they polymerize, they form this fiber that is called the F-actin or the fibrous actin. Similarly, this is the alpha helical region of the intermediate filament monomer. It becomes dimer, then it becomes the tetramer, and finally it forms the intermediate filament. Similarly, the microtubule is a heterodimer, and they arrange in a circle to make the tube-like structures, which is hollow at the center. And these are the places in the cytoplasm where the cytoskeletal elements are found. These are microtubuli usually found in the centriole, centrosome, flagella. Intermediate filament is distributed across the plasma, across the cytoplasm, and microfilament is found within the plasma membrane. So this is the functions of the intermediate filaments. We will discuss in the following slides that keratin filament is involved in the cell junction in epithelial cells, whereas in the connective tissue muscles in the glial cells, the type of the intermediate filaments is the bimentin and the bimentin related filament. And in case of the neurons, these are the neurofilaments. And nucleus is always, it is a nuclear lamine in all animal cells. So based on which cytoskeletal element is involved, the anchoring junction is of the following types. When it is actin and it is cell-cell junction, it is called the Adherens junction. When it is cell and extracellular matrix, it is focal. When the intermediate filament is involved, it is cell-cell junction, it is desmosomes, and when it is cell extracellular matrix, it is the hemidesmosomes. This is a type of Adherens junctions. So here we can see the Cadherin molecules that tethered inside the cell through the linker protein into the actin filament. It forms a continuous adhesion belt around each of the interacting epithelial cells, and it is found just below the tight junction in the apical end of the cell. So this network of actin filament can contract, giving the epithelial sheet the capacity to develop tension to change its shape in remarkable ways. By shrinking the apical surface of an epithelial sheet along one axis, the sheet can roll itself into a tube. By shrinking at the apical surface along the all axis, it invaginates into a cup, eventually create a spherical vacuole that is pinching up from the rest of the epithelium. This is used in the developmental biology during the embryo development. The neural tube is formed like this, that finally leads into the formation of the central nervous system and lens vesicles found in the lens of the eyes. Desmosomes looks like this. This is the cell 1, this is the cell 2. This is the cadherin, as we have discussed earlier. This is the linker protein. And the cytoskeletal element, that is cytoskeletal fiber, is the keratin filament. This is the intermediate filament in case of the desmosomes. So these are the rope-like keratin filament crisscross the cytoplasm. These are the spot welded, the desmosome junction, and it provides the tensile strength to the epithelium that is found in the epidermis. When it is, this is the keratin filament, this is the cadherin. When they are attached to the laminin found in the connective tissue of the basal lamina, this is called the hemidesmosomes. It is hemi because of it is half like the desmosome, that's why it is hemidesmosomes. Then we can see this electron microscopic figure that there is a gap between the two cells. And in this gap, there is the tubes, you can see. The channels, those are almost 1.5 nanometer in diameter. And the width of this gap is almost 2.4 nanometers. And the cytoplasm of the two cells, they interact with each other, they communicate with each other through these connections. And this gap junction, in this, along with this connection, there the ion channels are also involved that connect the cytoplasm of one cell into the another cells. Because of this, there is the electrical and the metabolic coupling. During the heart contraction, we can see that the cardiomyocytes connect with each other through this gap junction. So till now, whatever junctions we have discussed, it is summarized here. These are tight junctions. It seals neighboring cells so that nothing can pass across it. And it gives the polarity to the epithelium, the apical end and the basal end, so the apical surface or the basal surface. Then based on what cytoskeleton element is involved, the anchoring junction, it is of Adherens type, or it can be called a focalization, depending on it is cell-cell interactions or the cell and extracellular matrix, the cell-basal membrane interactions. Then if it is an intermediate filament, if it is cell-cell, it is called the desmosomes. And if it is cell and the basal lamina or the connective tissue, it is the hemidesmosomes. And when there is nothing in the between the cells, which is 2 to 4 nanometer in gap, and this is connected by the connections of the ion channel, this is called the gap junctions. Along with this type of junction, there is another type of junction that is called the synapse, where the one neuron that innervate or that makes a connection with another neuron, muscle cells, or a gland cells by junctions called the synapse. In this case, there is a secretory vesicles that secretes from the axon terminals by the secretory vesicles, like the neurotransmitter acetylcholine into the synaptic space, and that goes and binds to the receptor found in the receiving end. This is called the synapse. So these all junctions are in vertebrates. So in invertebrates, we have a counterparts of the tight junction that is called the septate junction. And there is another type of the junction that is called the apical junction. That is invertebrate specific. So the proteins involved in the tight junction in case of vertebrate is the Caludin and the Occludin. And in case of the invertebrate, it is the neurexin. So whatever junctions we have discussed till now, they are all found in animals. And these are absent in the plants because plants have a cell wall and it provides the strength and it holds the cells together. We have discussed the Plasmodesmata in the ultrastructure that connect the cytoplasm of the different plant cells. That is counterpart of the gap junctions found in the animal cell junctions. Through this Plasmodesmata, there is the inorganic small molecules movement As well as the macromolecules like the protein and the regulatory RNAs, they move from one cell to another. Like this, all the cytoplasm of all the cells found in the plants are connected to the plasmodesmata. Thank you.